All right, so proteins, I'll put over here, proteins are made of various small but similar in a way, similar building blocks. All right, and what are these building blocks? They're going to be called amino acids. So they're going to be 20 different ones, but why similar? Because well, they have to be an amino acid. Okay, it cannot be a monosaccharide or something like that. So it has to be a amino acid. That's what makes them similar. But there are differences between amino acids. So an amino acid is going to be a monomer. Okay, the monomer of proteins contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Like I said, there's not just one type of amino acid. There are different amino acids. So they're going to be 20 different amino acids. And that's all you need. 20 different amino acids. Of course, we may have a lot of repetition, but that's all you need to make all your proteins that sustain your life. An amino acid consists of so let's talk about the amino acid itself, just by itself. Consists of something called a central carbon, which is going to be this one here, a central carbon in the middle, that is going to be covalently bonded to a carboxyl group, an amino group, an amino group, a hydrogen atom, and your side chain, which is your R group, which is a very important part of the amino acid. The R group is really what gives the amino acid its special chemical properties. So, in other words, all amino acids are going to have a carboxyl group, an amino group, and a hydrogen. However, this is the only thing that kind of changes between different amino acids. For example, here you have two examples. We have leucine and serine. And we know that we can see here that this is the R group. This is the R group here. Okay. You see how this R group is different from this R group. Okay. So again, uh, amino acids, the what makes each amino acid different is going to be this side chain, also known as an R group. So what are the why are our groups different? Well, it gives them special chemical properties. Some of them may be polar, okay, which means they interact with water. Some of them are non-polar, okay, which means that they're going to be shying away from water or hiding from water. Some of them may be charged or ionized. In other words, they may either be positive or they may be negative. Okay. So this is only about the R groups. All right. Some of them are going to be polar, non-polar, charged, ionized, positive or negative. And these R groups are going to dictate how the protein is going to fold. And this is extremely important because, as I'll talk about it in a few slides, the shape of the protein determines the function of the protein. Okay, so I'm going to put here a little note here. The interactions between different R groups determines or determine the overall shape of a protein. Okay, and again, the shape is going to lead to its function. So so now that we know the amino acid, the monomer, part of a protein. Now let's see how we can get them together to make a larger chain. So amino acids are going to be linked together by something called a peptide bond or peptide bonds. Peptide bonds, which are covalent bonds, and bonds, which are covalent bonds. And these are, whenever you hear peptide bonds, that means it's going to be between two amino acids because we're making a polypeptide. How do we make this bond? How do we create these peptide bonds? Via dehydration reactions. Dehydration reactions. Okay, and that's what you see over here at dehydration reactions. Again, we lose water, but we make a bond just over here. And this bond is called a peptide bond. Okay, now we have two, one, and two. Oh, let me fix that a little bit. Now we have two amino acids together. Okay, one and two. Two amino acids are together. Now they are connected by a peptide bond. The proteins or the peptide chain eventually, and I'm actually going to be, I want to be a little more specific. So let's just say that amino acids are linked together by peptide bonds. The goal, forming a polymer. And this polymer is going to be called a polypeptide. Polypeptide. Polypeptide is a chain of amino acids. This polypeptide is going to have an amino end, which is an N terminus, okay, something like this, ends up with a nitrogen at the end or N terminus and an amino end. The other end is going to end up with a carboxyl end. We call that the C terminus. So the end of the chain is going to look like this. And proteins can be of several thousand amino acids long. Some of them can be smaller. Some of them are going to be way larger. Okay. And this is something that I want you to kind of understand. These are different amino acids, amino acid 1 and 2 and 3 and 4 and 5. Okay. All connected together. Okay. And let me see, 5 amino acids connected together. Okay, and it's going to form a polypeptide. The polymer is going to be a polypeptide. Keep that in mind.